Okay, well, I'm back again at the same alternate station. So again, you're still not going to see me today, but I did get some news that I think is worth sharing on something we've been following pretty closely. So we want to take a look. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for over 40 years, and I am recovering from cancer surgery, but that kicked off a whole MS thing. Who knows why? But at any rate, I'm not really walking right now, and my optic nerve's a little funky, so I'm doing the best I can, and I really appreciate it. Last week I got some encouraging comments from several of you, and I do appreciate that. I'm going to try to keep this going in the less than ideal way until I can get back in my chair and do things as I'm used to doing them. But this news has to be shared because it's timely and it's something we've all been following and pretty interested in. So why don't we go ahead and take a look at this latest clinical trial? I suppose before we dive into the clinical trial, we ought to remind ourselves of why we're excited or guardedly excited about Frexalimab in the first place. I did post a video, it's been about a year and a half now, about Frexalimab, and in that video I featured a post from Dr. Gavin Giovannoni, otherwise known as Professor G, and he talks about why he's excited about Frexalimab. Let's go ahead and listen to that, and then we'll look at the clinical trial.
I'm incredibly excited by this publication, and I think it's enormous or very big news, and it actually has rejuvenated my enthusiasm for the future of MS treatments. And the results are incredibly positive. When you look at the high dose, it reduced MRI activity by 90%, which puts it in the very highly effective therapy bracket. And over time, their therapy got even better. And this trial was very short. It was essentially a 3-month trial, simply because it's becoming increasingly difficult to put people into placebo-controlled trials for longer than this period of time. So I think the results are incredibly important. And not only did it have an impact on MRI activity, but it also reduced blood neurofilament levels and an inflammatory marker called CXCL13 quite profoundly in a very short period of time. And it also had an impact on quality of life. So this is remarkable results for such a short study. I also think that going beyond just the results is telling us something really important about the pathogenesis of MS, and it confirms the critical role of T cells in the pathogenesis of MS, because this particular pathway is critical in activating T cells. And so it's suggesting that MS is an autoimmune disease. And the problem I have, and it causes cognitive dissonance, is that I've taken a very strong position on EBV being the cause of MS. And if EBV is the cause of MS, how does this trial result with an anti-CD40 ligand explain Epstein-Barr virus driving MS disease activity? I'll also take the position, even if I'm wrong about EBV and Frexalimab, the results of this study are sending a real buzz through the MS community, particularly amongst immunologists, and actually also genomics experts, because both the immunologists and the genomics experts predicted that the CD40 ligand is critical in MS pathogenesis and predicted these results 2 decades ago. Now, the reason why the first generation of monoclonal antibodies against CD40 and CD40 ligand failed is because they tended to activate platelets and cause thrombosis. We now know about that, and it's not to do with it's to do with the what we call the FC fragment of immunoglobulin, and by engineering out the platelet binding domain of those antibodies, you actually prevent that from happening. So this new generation of anti-CD40 ligand antibodies, including Frexalimab, have not got thrombotic complications, which is reassuring. So this drug is not going to be, in my opinion, associated with thrombosis. Anyway, I was asked by a colleague yesterday when these results came out, where do I see the MS market in 10 years' time? He doesn't think there's space in the market for another anti-inflammatory like Frexalimab. And I think, his opinion is that because of the stunning results of the anti-CD20, their relative safety, ease of use, that there is no space for another anti-inflammatory. And so I disagree with him, and I suggest that he read my extensive commentaries on anti-CD20 therapies. It's very clear that anti-CD20s are very effective at suppressing relapses and focal MRI activity. In other words, you become inactive with no evident inflammatory disease activity, but it doesn't stop. These therapies do not stop smoldering MS. So a significant proportion of people on anti-CD20s will continue to get worse because it's not targeting CNS mechanisms and what's driving smoldering MS. And also, I don't think people can stay on anti-CD20 therapies lifelong. A proportion develop hypergammaglobulinemia, the immunoglobulin levels drop. And as you age, the risk of infection increases. And we are seeing an increasing number of patients being admitted with severe infections. So we definitely need an exit strategy from anti-CD20 therapies. And Frexalimab may be one of those exit strategies. And so if the phase 3 Frexalimab studies show the drug to have a much more pronounced effect on the end organ, in other words, reducing brain volume loss, people develop fewer paramagnetic rim lesions, fewer slowly expanding lesions. There's a reduction in spinal fluid biomarkers of both B and plasma cell activity, less microglial activation. Then Frexalimab will win hands down. Who wouldn't want to go on to the therapy that reduces or prevents smoldering disease? So I think this issue about is there space will depend on how good Frexalimab proves to be in phase 3 trials. So we shouldn't second guess that. And there's also this very tantalizing possibility that Frexalimab may reestablish immune tolerance, and it won't need to be given continuously. So there is this theory that Frexalimab and this class of drugs may reestablish immune tolerance and switch off autoimmunity. In other words, cure MS. Get rid of the autoreactive T cells. So I think we should wait to see what the results are, and the idea that you have to stay on Frexalimab lifelong may not be necessary, because if it does reestablish tolerance, you may only need a short course of it. It's clear to me, and it should be clear to my colleague and my colleagues in general, that anti-CD20 therapies have a limited shelf life as a treatment for MS in the way we use them now. And I don't think they will be the mainstream of MS treatment in 10 years' time. And you may not be aware, but all the big pharma companies that have licensed anti-CD20 therapies are not sitting on their hands. They all have very deep and very interesting MS development pipelines. And this tells us from within the pharmaceutical companies that have these therapies that they are not good enough. And and it's clear that we have to go beyond just preventing relapses and suppressing focal MRI activity. And, we have to get to the smoldering MS. And so the most exciting of these products at the moment are the CNS penetrant therapies targeting smoldering MS, and this includes CD19 targeted CAR T cells and proteasome inhibitors like ixazomib.
And here is the clinicaltrials.gov page. The name of the study is Efficacy and Safety Study of Frexalimab in Adults with Non-Relapsing Secondary Progressive Multiple Sclerosis. And as you can see here, the sponsor is Sanofi, and they are the ones providing the information. The last update was posted in August of 2025, so this is brand spanking new. And the study overview says that the purpose of this randomized, double-blind, placebo-controlled parallel group study is to determine the efficacy of Frexalimab in delaying the disability progression and the safety up to 36 months double-blind administration of study intervention compared to placebo in male and female participants with non-relapsing secondary progressive MS aged 18 to 60 years at the time of enrollment. People diagnosed with non-relapsing SPMS are eligible for enrollment as long as they meet all of the inclusion criteria and none of the exclusion criteria. And now we are going to see what some of the details are. First, this event-driven study will end when the target number of 6-month CCDP events is achieved, and I could not find what CCDP stands for, but I think we get the general gist. They're going to keep going until they get what they think they need to get. And the study is expected to last 43 months from randomization of the first participant to the common study end. And then secondly, the number of scheduled visits will be up to 25, including 3 follow-up visits with a visit frequency of every month for the first 6 months and then every 3 months. And under the contacts, I notice they don't give the name of a particular person, but they do give you a toll-free number to call, and there is an email at Sanofi. You can just contact them if you want to email them for more information. I think what's really impressive here is that the study has 321 locations. And I'm not going to go through these in detail, but I'll just scroll through this. And in the United States alone, you can see that pretty much every state is represented here. And let's see. I'm just going to scroll and let this play, and you can see yourself that your state is probably represented here if you live in the United States. And of course, you can go out to the website and look for yourself to if you see anything you want to explore a little further. And now, let's what's really exciting is how many countries outside the United States are also included. We've got Argentina and Australia, which I know a number of you will be excited to see because they don't apparently do an awful lot of these trials in Australia, but I know a number of you are from there. So there's several locations in Australia and Belgium and Brazil, lots in Brazil, Bulgaria, Canada, Chile, China, Czechia, France, Germany, Greece, Hungary, India, Italy, Japan, South Korea, the Netherlands, Poland, Portugal, Puerto Rico, Spain, Sweden, Turkey, Ukraine, United Kingdom, and there you go. That is pretty neat that we have so many outside the United States. If you can't find a test location that's close to you, I'd be very surprised. But of course, we can't get too excited until we see what the eligibility criteria are, both for the inclusion and the exclusion. So let's just go through those. Inclusion criteria, which means if all of these apply to you, you could be included in the study. The participant must have a previous diagnosis of relapsing remitting MS in accordance with the 2017 revised McDonald criteria. Participant must have a current diagnosis of secondary progressive SPMS in accordance with the clinical course criteria revised in 2013, endorsed by an adjudication committee. The participant must have documented evidence of disability progression observed during the 12 months before screening. Eligibility will be analyzed by an adjudication committee. There needs to be an absence of clinical relapses for at least 24 months. The participant must have an EDSS score at screening from 3.0 to 6.5 points inclusive. And then the next one is contraceptive use by men and women should be consistent with local regulations regarding the methods of contraception for those participating in clinical studies. So I guess you'd have to find out more about that, because I'm not really sure what that said, all said and done. And then finally, for patients eligible to be treated with siponimod, 1. does not tolerate it due to side effects or safety reasons, or 2. has failed siponimod treatment due to perceived lack of efficacy, or 3. has declined siponimod treatment. Okay, so then moving right along, if you made it through the inclusion criteria, here are the exclusion criteria. The participant has a history of infection or may be at risk for infection, the presence of psychiatric disturbance or substance abuse, history, clinical evidence, suspicion, or significant risk for thromboembolic events, as well as myocardial infarction, stroke, and/or antiphospholipid syndrome, and any participants requiring antithrombotic treatment. And I think if you're affected by any of that, you would know, or you could certainly check with your doctor. The next one is history or current. Oh my goodness, these words are long. Hypogammaglobulinemia. Hypogammaglobulinemia defined by values below the lower limit of normal, LLN. A history or presence of disease that can mimic MS symptoms, such as, but not limited to, neuromyelitis optica spectrum disorder, systemic lupus erythematosus, Sjogren's syndrome, I know I'm pronouncing that wrong, acute disseminated encephalomyelitis, and myasthenia gravis. The participant has sensitivity to any of the study interventions or components thereof, or has a drug or other allergy that, in the opinion of the investigator, contraindicates participation in the study. And then finally, the participant was previously exposed to Frexalimab. So there you go. That's all the reasons you might not be eligible for the study. But if all of those seem to check the right boxes for you, go ahead, if you like, and find out more at the contact information that we showed you above. Well, there you go. I think now we're all caught up on the latest with Frexalimab, and I will keep you posted as test results become available. If you are interested in learning more or perhaps being a part of the clinical trial, you saw that there was some contact information there. And I would advise you to think carefully about the clinical trial as you would any clinical trial or any drug treatment or any other intervention that you decide to take on. What are the risks? What are the downsides? This one sounds pretty good, but you'd really want to ask a lot of questions because your own body is unique and it might have sensitivities that others don't, and you want to be careful of that. But in either case, we are looking forward to seeing something good come out of this because, as Professor G says, it looks like this might even be not only an improvement in the course of our disease, but a possible cure for it. And boy, you sure don't hear the word cure very often. I like the sound of that. Okay, well, whatever you decide to do, I really want you to take good care of yourself and stay in as top condition as you possibly can. These new developments are coming thick and fast, and there's also lifestyle interventions we're learning about, any one of which could help make our quality of life so much better. So let's be in the best of health we can be to take advantage of those. In the meantime, take care of yourself, and I'll see you again in my next video.